final control plan for Bloomkin fever is a presentation on the workings of the research team comprised of Corin Dunn, Austin Heron, Abigail Malthner, Carrie Bolger, Keshava Gali, Vivian Escalon, and Jesus Garcia. Bloomkin's fever. Our team was tasked with identifying and stopping a recent outbreak of an unidentified vector-borne disease. To do this, our teams first devised a plan to collect data about the disease through surveying patients and learning accounts of the patients, as well as using the sites of incident to construct a map of affected areas. Then, through that information, we hope to identify potential vectors of the pathogen, determine the disease agent, and finally to devise and enact a plan to stop the spread of the disease now known as Bloomkin's fever. Our task proves vital for the sake of current victims of this disease, preventing the further spread of Bloomkin fever, and to ensure public health and safety. Patient and data collection. 10 patients originally presented symptoms and had test results that indicated infection by an unidentified vector-borne disease. Our team first collected information on patients by issuing a 28-question survey. The purpose of this survey was to gather information about how exactly these people came into contact with the vector and disease agent. The question proving most vital to our investigation was contact with mosquitoes and recent bites that might have occurred. Background information was then obtained on the patients and used to determine the likely source of outbreak. Using the collected data, investigators were able to determine patient 0. It was identified to be Dr. Jeff Bloomkin. Dr. Bloomkin came into contact with the disease through rats he had recently imported from China. He then traveled to the other sites of infection, carrying the disease and distributing the infected rats as gift or through transactions. A map was also generated based on area of incidence, and it appeared that susceptible areas were those around the Gulf Coast, from coastal Texas around the Galveston area to Miami, Florida. The sites of all originally reported cases can be found on this map and are indicated by a gold star.
Our vector is found to be Anopheles bradley. It was discovered by G. H. Bradley and W. V. King in 1939 and belongs to the genus Anopheles, which was originally classified in 1901. Some distinguishing characteristics of Anopheles bradley are that its maxillary palps are equal in size to its proboscis, which is a key characteristic in Anopheles species. The mosquito also positions its abdomen parallel to its proboscis when feeding, as opposed to other adult mosquitoes which may position their abdomen parallel to the surface it is feeding on. But the most distinguishing characteristic of bradley is the white pale scale spots and pale dots on their wing veins. Anopheles bradley also has a wide geographic range residing across the southeastern states and the northeastern coast. Although it possesses a wide range, its highest concentrations are found in Texas, Louisiana, and Florida. Like all mosquitoes, the female adult requires a blood meal in order to complete their reproduction cycle. The proteins in the blood are not only act as a source of energy for the mosquito, but are also vital for the maturation of the ovaries. Females prefer mammals, including humans, and transmit the disease during feeding through their salivary glands where their disease resides.
Familiar with this little guy? He's popularly known by the medical community for vectoring a variety of diseases. One of his contributions was playing a large role in spreading a little pandemic called the bubonic plague. The roof rat, or usually known as the house rat, is a common long-tailed rat of variety shades of brown or black. This rat is primarily nocturnal and can adapt to a variety of habitats, both urban and rural. Its peak breeding seasons tend to be during the spring and fall. The Anopheles bradley mosquito, our vector, and the roof rat share common geographical ranges. If Anopheles bradley is needing a blood meal, the roof rat is an excellent source to feed on. Not only are they easy to find, but these creatures are so adorable they're hard to resist. Little does the Anopheles bradley mosquito know, the itty-bitty roof rat is a reservoir for Bloomkin fever. This means Anopheles bradley could potentially spread Bloomkin fever to innocent humans when it needs another blood feeding, because we too are oh so adorable. Well, most of us. Here we have another adorable little creature. This mouse hits a little closer to home because it may very well live in your home. No need to clean up after a meal when you can just have the mouse gladly feed on your leftovers at night while you sleep thanks to its nocturnal behavior. These furry little guys live just about everywhere in the world except for most of South America, Africa, or places where it gets too cold for their tiny little bodies. Luckily for us, North America fits all their needs to call our entire continent home. The house mouse is connected to Anopheles bradley, similarly to the roof rat, by simply living in the same places. If you have a roommate, you're bound to run into them on a frequent basis, unfortunately even if you aren't besties for the resties. What's even more convenient, though, is the house mouse and the Anopheles bradley enjoy humans. So Anopheles bradley can get a little blood meal from the adorable house mouse, again completely oblivious to the fact that such a cute creature could be the reservoir for such a nasty disease like Bloomkin fever. And then, just around the corner, there's a yummy human being who will provide another blood meal, and in the process, Anopheles bradley will generously share the disease with its human host.
Proteus astros, the disease agent of Bloomkin fever, is a flagellated gram-negative bacterium with a genome size of 4.49 millions of base pairs. This bacterium tested positive in both the indole and catalase tests. Also, it ferments many sugars such as maltose, mannose, lactose, and glucose, and produces a gas upon fermentation of salicin. Growing conditions are best for Proteus astros in temperatures between 37 to 40 degrees Celsius. Bloomkin fever is a deadly form of meningitis. Meningitis is the inflammation of the meninges which cover the brain and spinal cord. This type of disease is most commonly spread through infected airborne respiratory droplets via coughing or sneezing. The most prominent symptoms associated with Bloomkin fever and meningitis in general are neck stiffness, persistent headache, and fever. In order to suppress these symptoms and hopefully treat the disease itself, antibiotics are often given. Unfortunately, these do not always stop the disease, and sometimes supportive care is the only treatment available to those infected. If left untreated, or if the disease does become untreatable, patients may be left with permanent brain damage or learning disabilities. Those most commonly affected by this are babies, small children, the elderly, and those who have weakened immune systems.
Our control plan consists of several different breaks in the links of the disease cycle. Eliminating the disease agent is the first step. The Proteus species can be found in drinking water. When they are ingested, they become an unwanted organism in the intestinal microflora. Presence of the organism is usually associated with fecal contamination. Because of this, one way to eliminate it would be to increase the number of wastewater treatment plants in the region. This would reduce the amount of fecal coliforms of the Proteus astros. Since mosquito larvae feed on this organic matter, eliminating fecal coliforms would in turn help eliminate Proteus astros from the environment. For controlling the vector, previous use of commercial pesticides has led to a controversial response. Many residents did not like having chemicals sprayed near their houses. To substitute, we will implement genetically modified male mosquitoes. Since mosquitoes mate for life, releasing sterile male mosquitoes will reduce the overall population over time. Also, removal of standing water in communities will eliminate breeding area for these mosquitoes.
Okay, so we plan to control these reservoirs by many means. For example, the specific vector disease targets two reservoirs, which are human and rats. Our plan to control the human reservoir is to alert the masses by means of social media as the primary source to get the news to people. Titling, "We have a new disease in your area and it's about to kill you." So here's what to do. The second one is pretty self-explanatory, which is go to the doctor and get your regular checkups. Have some common sense. Stay away from stagnant water, and if heading outdoors where a report of an outbreak have been cited, please wear protective gear such as long pants and sleeve shirts. with little bug repellent just in case. Also, don't forget to report any kind of bites, which are such as rats or mosquitoes. Lastly, the rat reservoir, we will have to catch them, trap them by any means, and test them to see if they have any type of trace of the disease. If the reports come back positive, the last thing we'll have to do is to eradicate them. Okay, so if you don't like our last plan, eradicating all the mice and rats, we can always take another route. And that we can take, if we don't want to kill them off, will be, because they're so cute, we can always vaccinate them. Since the disease is a protease, it can be cured by means of antibiotics. We don't need to vaccinate every single one, just enough to create a herd immunity. This will stop the spread of the disease agent.
After implementation of the control plan, we will begin the monitoring phase. Community-wide surveys will be given to see if residents have noticed changes in mosquito presence. In addition, the collection of adult mosquitoes and mosquito eggs will be done to determine if the mosquito population has increased or decreased. By surveying hospitals in the affected regions, prevalence of Bloomkin fever will be determined to see if there was an increase or decrease as well. The effectiveness of our control plan will determine if changes to the protocol are necessary.
Okay, now to wrap things up, you must have heard all this information, and it must be overwhelming. But it's good for you, because it will keep you alive. Anytime a link is broken in the cycle of vector-borne diseases, it reduces the ability of the pathogen to successfully complete the disease cycle and further spread the disease. While cases of Bloomkin fever have not reached a staggering figure yet, if actions were not to be taken, the situation could quickly evolve into a devastating outbreak. Thank you for watching.